Hi there guys, and welcome to another A-level biology video. Mr. Phillips, and today we're looking at receptors and how the receptors are part of the reflex arc and how they start to stimulate that action potential, which is the nervous impulse. So let's have a look at it. So we've talked about this before. Receptors are specialized cells that detect one specific stimuli and a change in that stimuli. And a stimuli is normally an energy change, such as temperature increase or light change, or as we're going to look at today, pressure potentially. And basically what they will do is they'll convert the energy from the stimuli into an electrical impulse. And often they could be a specialized cell, or they could just directly be a part of the sensory neuron, which then obviously stimulates the reflex arc directly. So receptors, basically, you need to understand that, as we said, they are specialized cells. They detect a specific stimulus, which is an energy change, and as a result, they are what we call energy transducers. And what this means is that they change or convert one type of energy into another. So they normally convert the stimulus energy into the electrical energy. And there are lots of different types of receptors that you might be able to talk about. So there's rods and cones, which are in the retina, and they stimulate the light energy is the input energy stimulus, and they turn it into electrical energy. Then you've got olfactory lining in the nose, which is a type of chemical energy, which again is the output becomes electrical energy. You've got pressure receptors, which is the input energy is movement, which again turns into electrical energy. You've got the ear, which is sound energy, and that again turns into electrical energy. And muscle spindles, which might seem like a strange receptor, but if it detects the movement of your muscles, that could then trigger the electrical impulse, which makes your brain aware of what's happening. So one of the key things you need to learn, one of the receptor examples you need to learn, is a Pacinian corpuscle. And this is basically a layer of protective tissues around an axon of a sensory neuron, and it's a very common type of receptor that is used to detect touch, basically, because it detects pressure. It's sometimes called a capsule, and we're going to be talking about the membrane being lamella, which you'll have heard of before. Obviously, we've talked about lamella in membranes in the chloroplast before, so we're going to be using the word lamella as the membranes. So this is like the standard receptor that you must remember. As I said, it stimulates all our touch, and it goes directly onto the sensory neuron. So this neuron here is in fact your sensory neuron, and the Pacinian corpuscle is at the end of the sensory neuron with this layer of membrane wrapped around it. So it's often found in the skin dermis, so basically where you feel your touch. It contains endings of the sensory neuron, as I said, and layers of connective tissue wrapped around it called lamellae. And when you apply pressure to this, so let's say I'm applying, we'll see this better in the next slide, but when I apply pressure to this, it basically causes that lamella to push in onto that lamella, which pushes into that lamella, and so on, until you get to the actual nerve, and it causes a deformation of the nerve on the inside. Now, this is where this module is quite difficult to teach in the right order, because if you know about action potentials, you'd know how the nerve impulse is fired. Basically, a nerve impulse is fired by opening sodium channels, which allow sodium to rush in and out of the nerve cell. And by doing this, pushing on it, it deforms the sodium channels, causing them to open, and that triggers the threshold for a nerve impulse. So the key thing to remember at the minute, and you might need to come back to this once you've learned about nervous impulses, is it causes the sodium gates to open, allowing sodium to flood in, and that depolarizes the membrane. Again, you'll understand what that word means later on. It means the charge of the membrane changes. So what we call these are stretch-mediated sodium ion channels. So they're triggered by the pressure on the lamellae of the Pacinian corpuscle, which opens the channels, and that triggers your impulse. So effectively, if you look at it from here, this shows you again stuff that you'll learn a bit more detail coming up. The resting potential, we've got the nerve impulse, sorry, the sensory neuron here has got a positive charge on the outside, negative charge on the inside, so we call it negative the resting potential. And there's no sodium ions being opened at this point, so sodium is these positive charges all on the outside. but then when pressure is applied, that causes the sodium ions or channels, the stretch mediated channels, to open, and that means the sodium then can rush in to the sensory neuron, and that changes the charge on the sensory neurons. It becomes positive on the inside, and as you will find soon, that is what causes a action potential. And what we say is it reaches a charge where it becomes a generator potential, and it gets positive enough to cause a nervous impulse. Now, what we also need to know about then is the vision receptors in the retina as a specific example. So the retina is obviously part of the eye, and the amount of light that enters the eye is controlled by the iris, which opens and closes your pupils effectively at the front of the eye. And basically the receptor cells contain optical pigments which break down when the light is shone onto them. So as the light hits the retina, obviously it'll be a straight line, not like I'm drawing now, okay, that line causes the breakdown of pigments in the light, and that then triggers the action potential, which allows us to see things. And effectively we have this area here called the fovea, and that is a particular area where we get most of our vision because it's an area that has loads and loads of photoreceptors. And basically once the photoreceptors are broken down, that triggers a nerve impulse, when the nerve impulse is carried to the brain via the optic nerve. And where the optic nerve actually leaves the eye is called a blind spot. Okay, so you see that bit labelled here, and the blind spot basically is where there are no receptor cells, because that's where all the what we're going to call ganglions in a minute converge to form the optic nerve. And effectively how it works is when you have these photoreceptor cells, light hits the photoreceptor cells, and in there you've got light sensitive optical pigments, and effectively these pigments are bleached by the light. It causes a chemical change, and that then alters the permeability of the membrane again to sodium ions. So a bit like the Pacinian corpuscle, which pressure changed the sodium ion permeability, this bleaching of the pigment increases the permeability to sodium ions. That then creates an action potential. It reaches this generator potential, this threshold, which we'll talk about in the next few lessons, and then that sends then the nerve impulse along the optic nerve and to the brain. Now, there are two types of cells that we need to know about. There are rod cells and cone cells. So if we start first of all with the rod cells, there are about 125 million rod cells in your eyes, and these are very, very sensitive to low levels of light. So they're very useful for us to distinguish between light and dark levels, basically, and very dim levels. So this is really quite useful for us because it allows us to distinguish different levels of light. And basically one photon of light can cause an action potential, a nerve impulse in rod cells. But they're, as a result, because they're so sensitive to light levels, they're not very good at sensing different colours. Whereas cone cells, there are less of them in our eyes, and effectively these are more active during high light levels. So effectively they're less sensitive to actually light, but they're very sensitive to different wavelengths. So these are ones you tend to use during the day, which is why you can see more vivid colours during the day than at night time, which is when your rod cells work. And there are three types of rod cells with different pigments that bleach at different wavelengths of light, and they're sensitive to blue, red, green wavelengths. And when we see all the different colours, it's because they combine and they're affected different by different wavelengths, and that provides us with our colours that we recognise. And this is called trichromatic vision. So the idea that we have three different types of cones, each with different optical pigments, and they're stimulated by the different proportions of the different wavelengths that your eyes are receiving. So let's have a look at these in a bit more detail. So here we have a picture of the rod and cone, and it shows you that the rods and cones are not directly linked to the actual optic nerve themselves, or to the central nervous system. they're actually connected by cells called ganglion cells, and these cells then connect to the optic nerve, which then connects to the brain. And also within the eye, they're not even evenly distributed. So what we have here is you have most of your cone cells in this area, which is your fovea. So apologies if you can't see that, probably shows a bad colour there. So most of the cone cells are found in the fovea, where most of the light focuses, and that gives you the colour reception you can see, whereas the rod cells tend to be found around the outside of that, which is where you just detect the light, but you don't need that colour detection so much. Now what you can see here is the difference, if you look at the picture on the left here, of the rod and cone cells and how they're actually connected to the optic nerve. So what this also shows us though is why they have their different visual acuity. So rod cells are, as we know, very sensitive to light. There, as we said a minute ago, the ones that are found that work best in the dark, and they don't detect much colour, but they detect very small differences in light detection. And this is because of how they're set up. So if you look at it, to connect to the optic nerve, many rod cells connect through one ganglion cell to the optic nerve. So this does something called summation. So normally, let's say you have to get to this threshold level to fire a nerve impulse so the brain detects the light. Well, because many rod cells connect to just one ganglion, they basically can all be added together. So let's say each one of them had a score of, let's say, 10. That would be 10, 20, 30, 40, 50, 60, all added together onto that one. So this one would get 60 effectively, because it all comes together from those 6 different rod cells. Now this means that the rod cells can basically join together on the neuron, and many weak generator potentials therefore can combine to reach the threshold and trigger an action potential. However, it's different then with cones. Cones are less sensitive to rod cells because one cone basically normally joins to one individual bipolar neuron. So it will take much more light to trigger the action potential in the ganglion cells at the bottom. So basically this one doesn't have any extra light, it just goes all the way down into here. And if it's not enough light to trigger a nerve impulse, it won't fire that nerve impulse. Now, in terms of being able to see the differences in colour, this setup also shows you this as well. So as we know, rod cells are not very good for us to see colour. They're good at seeing light levels, and the reason is because they all join up. So if you imagine, let's say this rod cell here is the first one that I'm drawing the red blob on now. Let's say this one detected some red light, but this one next to it detected blue light, and then this one next to it detected green light. Well, the problem with that is that's very good well if they're all connected to different nerves to the brain, but they all merge together, and because they merge together, therefore it makes it very difficult for us to see light. So effectively, visual acuity, which is the ability to tell apart points that are close together. Rods give us low visual acuity because many rods join to the same bipolar neuron or same ganglion, and that means the light from the 2 points can't be told apart. So it makes it very difficult to see those differences. Whereas cones have a high visual acuity because the cones are close together, but they only have one neuron. So basically, when you hit 2 cones, 2 action potentials go to the brain, and they are detected as sort of separate entities, which allows us to detect it. So think about the word resolution at earlier on in the course. The higher you'd have a higher resolution, a higher visual acuity with the cone cells, because each individual cone cell is sending an individual message to the brain, and all of those individual messages can be interpreted differently as a result. So just to finish off then, so we said that light falls on the receptors, they send the trigger to the brain. each cone cell connects directly via one ganglion cell, whereas many rod cells connect directly via one ganglion, and therefore you've got less visual acuity from the rod cells, but you have a higher detection of light. Now, just to summarize then the difference between rod cells, that pretty much says it, but the rod cells, we've said, because of the many of them connect to one ganglion, they will be very sensitive to light levels, but often they will give information in black and white and give low visual acuity, whereas the cones have obviously one cone to one ganglion, and therefore they give lots of information in colour, high visual acuity, but they've got a low sensitivity to rod cells. So really important, guys, that you look at the stuff to do with light and make sure you've studied it in detail, because you would be expected to be able to describe the difference between light detection, visual acuity and colour detection in the rod and cone cells. So again, just to summarise, I think it's so important. Visual acuity is how far apart you can separate two spots of light can be seen as separate. So exactly the same as the term resolution for microscopes. And we said because each cone cell has its individual ganglion, each individual wave of light will be stimulated by the brain, whereas 100 cells of rod cells may have one ganglion. So 6 spots hit 6 different rod cells, they might only be detected as one spot by the brain, and that means your visual acuity is a lot less as a result. And I think that really, the other side of that is also we've got a summation point, okay, and that is that when, however, dark objects are going to be seen less clear, but we are going to be more sensitive to the actual light levels. So I hope that's been useful for you guys. Make sure you really do read up on rod and cone cells, because they're a big important part of the A-level course, and make sure you're happy with Piscine and corpuscle, and you can apply both of those to the action potential, which will come up in future lessons. Thank you very much. Hope it's been useful. See you soon.